Hello and welcome back. We'll be talking about DNA replication today. That's great. Nice to meet you. Okie dokie. We are going to talk about DNA replication. And we are going to... We are going to use the terms for prokaryotic DNA replication. Now, the names for the eukaryotic enzymes are different, but we don't care. We're just memorizing one of them. So we're choosing prokaryotic to use as our system. And we are going to use these. And then we're going to talk about the differences in replication between prokes and euks. And I'll give you a little hint. There's not much difference. As it turns out, it's really just the origin of replication that's different. So, that's a little foreshadowing. We'll get to that later. But for now, let's talk about the enzymes. So these things you have to memorize. Have to. We have to have a conversation, which means we have to know the vocabulary. So let's just go through these really quickly, and we'll be done. Great, thank you. The DNA polymerase, right? This is the enzyme that extends the primer that actually does complementary base pairing. DNA polymerase 3 is the guy that makes the new strands. DNA polymerase 2, this fills in where the primers used to be. Hold on to that. We'll get to it. Don't panic. Other enzymes we give a crap about is helicase. That's the one that breaks. What does it do? It breaks hydrogen bonds. Right? The only way we can unwind the double helix is to break it apart. What holds a double helix together? Yes, you guessed it. Hydrogen bonding.
Now, this is important.
Right? Thanks, Rocket. It is important. What about this? Single-stranded binding proteins keep the helix open. A common theme that I'll reiterate over and over is that if there are two strands of DNA or DNA and RNA or RNA and RNA and there is complementary base pairing on either strand, A's wanting to get together with T's, G's wanting to get together with C's, it will happen. The only way it won't happen is if we actually keep it apart. So in order to keep the two strands apart while we're trying to do DNA replication, we need single-stranded binding proteins. That's really great. Okay, so what the heck is going on here, people? What's the next one? Primase. That is the enzyme that creates the RNA primers and complementary base pairs them to the template. We need to know that. And then our good friend ligase, which links together the Okazaki fragments and any place there was a primer, which essentially means at the origin of replication between the Okazaki fragments. And when we say weld together, do we mean they make hydrogen bonds? No. Phosphodiesters. So if you didn't watch the tutorial about DNA, and if you didn't remember anything from your prereqs, you got to go back and listen to that, because you need to know the difference between a phosphodiester bond and a hydrogen bond, or you're not going to know what I'm asking when we get to those questions. When we see this chromosome, we see it's circular. What do you think that means? Yes, it's prokaryotic. Their chromosomes are in a circle. Ours, the euks, are linear. Like I alluded to earlier, one origin of replication per chromosome. What happens? Breaks open with the initiator proteins. Primers are added, and replication happens bidirectionally, which means it is progressing this way, and that way. So going this way and going that way till we get almost all the way around. When we finish it, boom, two circles. One old strand and one new strand. Semi-conservative. just like that other tutorial. Here's what it looks like under some sort of electron microscopy, just like the pictures they drew. Woohoo, there's where it's opening up, at the origin. Here's proceeding in both directions. Ooh, they're almost done. Oh, yay! 2 strands. Whoop-de-doo. What about euks? Here's something linear. Eukaryotic replication. There's lots of origin of replications. We like to call them this. Or actually, I do. I don't know if we do or not, but I do. So all we're saying is that it starts at multiple origins along a chromosome, and it proceeds in both directions from each origin. Opens up, proceeds this way, proceeds that way, this one proceeds this way, this one proceeds that way. At some point, right, they bump into each other, and there you go. You get all the way down the chromosome. Very nice. Yes.
Nobody talks to my friends like that.
What? I didn't say anything bad. I was just talking about bumping into each other. Jeez. Hey, remember what this is? Yes. It's a nucleotide. What kind is it? Deoxyribo. Why? Because it's deoxyed right there. Fascinating. Why do we care so much? Well, if we think about what we might have learned in our prereq, this looks a lot like ATP. What's ATP? The energy carrier. ATP, yes, ATP is a nucleotide. We call them DNTPs because they're deoxy, and the N stands for either A, or T, or G, or C. It's important that the nucleotides, or nucleosides, pardon me, carry their own energy, so then when we do replication, it's not a energy sink needing tons of ATP. We bring our own energy. When we cleave when we cleave the bond, we release energy. That energy is poured into creating that phosphodiester bond. It's so beautiful. And look, here it is, illustrated for us. The new strand being created in this direction, adding to 3' ends. That's the only way we can ever extend any new strand of any type of nucleic acid. Can only add to 3' ends, 5' phosphate, 3' OH. Bam! Cleave that bond right here. All kinds of energy. Dump that into creating that phosphodiester bond. It's really, really a beautiful thing. So,
I have a plan. You got a plan.
What?
Yes.
All right, this is my class, so just knock it off. I don't care what your plan is. Jeez. So that's just a picture. Ignore those guys. They're just annoying. I don't know why they can't just... Yes, we know. Okay, moving on. So what do we mean? Here's a nice little picture. It's not showing all the details, but it's giving you the main idea. Always, 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 strands of nucleotides, nucleic acids, are always antiparallel. Doesn't matter if it's DNA to DNA, DNA to RNA, RNA to RNA. Always, always, always antiparallel. What does that mean? 5' ends complementary to the 3' end, 3' end complementary to the 5' end. That's just how nucleotides fit together. Again, if you go back to the DNA chapter and look at that tutorial and look at the pictures, that's how it works. That's how, right, the hydrogen bonds between the two strands, phosphodiester bonds holding each nucleotide together within a strand. Showing the replication fork, it's opening this way. We're only adding to 3' ends, so this strand is going this way. Adding to 3' ends, this strand is going in towards the fork. Get it? Got it. What else? What else? Again, just showing us the same thing. This guy's going in. That guy's coming out, adding to 3' ends, adding to 3' ends. Have to do that. So if we were imagining here's the origin, one strand goes away from the origin to the fork, one from the fork to the origin. And so on and so forth. So if this is our origin, and this is where it's open to at time unit 1, and then we wait 30 seconds, I don't know, 1 second, 2 seconds, whatever. The fork open to here. This strand just keeps adding to this 3' end. Go, 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 doesn't matter, right? Now that was the first stop. Here was where we looked at the second stop. This guy just keeps adding. It's continuous because we can just add to the 3' ends. Well, what about the other strand? That poor guy, he has the 5' end here. He's going in this direction. So as the fork opens, right, we cannot extend. Why? Because the 5' end there, we can never add to 5' ends during replication for polymerase. We have to start a new strand, 5' to 3'. And then again, snapshot. So now here's the first one. Here's the second one. And now we have to start a new one, the third, and so on until we fall off the end. These are the discontinuous strands or the lagging strands. It takes longer, literally. The leading and the lagging. That's where those terms comes from. That's it. Well, what do you think of this? I think it's pretty dandy. What the heck? I don't know what the heck that was. It's a circle. What do you think that means? You guys are brilliant. I love it. Is there still a leading and lagging strand even though it's a circle? Yes. Do we have to be antiparallel? Yes. Can we only add to 3' ends? Yes. Huh. Well, there you go. So boom, there's our origin. Away from the origin into the fork, leading strand 3' end. Lagging strand goes towards the origin from the fork. Leading strand into the fork, lagging strand back to the fork. Huh. Whether it's a circle or a line, works exactly the same. What's different? Only one origin of replication in Prokes. Lots of origin of replications, or is it origins of replication? In linear chromosomes. See? I like what you've done here. Here's the origin, leading, lagging, toward the fork, toward the origin. It's beautiful, people. Okay, so here we're just showing the initiator proteins, the origin of replication. Not going to get all crazy with the protein stuff. Be ready for this in Biology 380, where they're all about proteins, we're all about DNA. But the initiator proteins binding the origin causes a short stretch of DNA to unwind. What do we mean by unwind? Broken H-bonds. Broken H-bonds. H-bonds, hydrogen bonds between pairs. Whew, sorry, I love complementary base pairing. Couldn't help myself. Then the single-stranded binding proteins. There's our little guys keeping this open. And then our good friend helicase hops on to open the forks in both directions, away from the origin. I'm getting teary-eyed. Yay! What does DNA gyrase do? It keeps it from getting totally tied up. If this is squeezing, opening, breaking hydrogen bonds, squeezing this, squeezing that, this supercoil stuff over here is going to get all jumbled up. So what happens? These guys go in front of the fork and they sort of break phosphodiester bonds, let it unravel, blah, and reattach phosphodiester bonds so that then this doesn't become all knotted up in a big mess, right? If this kept squishing and there wasn't a gyrase, this would end up going like this. And it's all squished up. You can't, like your headphones, right? If you have your earbuds on the stupid cord thing is always all jumbled up in a big giant mess. That would happen in our DNA. and then the forks would stall. We wouldn't be able to get by this. Replication couldn't happen. That would totally screw over mitosis, and you're all dead. Oh, sorry. Yeah, and dead is bad.
Nobody talks to my friends like that.
I know. I'm sorry, Drax. That was pretty rude. Okay, nobody's dead. Everything's fine because Gyrase exists, and it takes care of that super coiling and all the naughtiness. Everything's good, so don't panic. Whew. So here's the big picture. First, before anything else can happen, primase, our good friend, has to synthesize RNA primers and complementary base pair them to the appropriate place at the origin of replication or at the fork, depending on which strand you are, has to be antiparallel. Has to do complementary base pairing. Using those two rules, only add to 3' ends. With those rules, then you can't put them in the wrong place. If you put a primer at the origin and the 3' end is facing the origin of the primer, it can't go anywhere. You can't cross origin. Another rule. Don't cross origin, which we'll talk about later why you can't. It's not just a rule, but if you give yourself that rule, you can't screw it up. Don't cross the origin. Okay. Okay. So yeah, going back towards a primer on each. The continuous strand has a primer also. We can never start a new strand of DNA without a primer. They all need primers, whether it's lagging or leading. Lots of people forget that when they do their drawings. It's bad. Don't do that. All strands need primers. All right, what's this? Oh, it's a little table of crap you should know. What did we talk about? We talked about DNA polymerase 1, which we haven't gotten to yet. All we've talked about so far is DNA polymerase 3. That's the guy that's doing the adding to 3' ends, complementary base pairing off of the primers. Who made the primers? Primase. Don't forget, it's right in the name. Make a primer. It's primase. You got it. What else? Ooh, here's a little practice. This is very similar to my other tutorial, so I'm going to go quickly through this. Here's our origin of replication. Don't cross. If we look and we figure out which is the 5' end and which is the 3' end, and we need to be antiparallel, which just means, right, 5' to 3', that means the other strand needs to be 3' to 5'. So if we have a 5' here, that means we'd want the 3' end to be the other strand. So that would make this the 3' end and that the 5' end. And if that's our little RNA primer at the origin, then DNA polymerase 3 can grab onto that 3' OH and complementary base pair. Because we know that the other strand is going to be the opposite, 5' to 3'. There's our RNA primer, and that works, yes? Because when we take this strand and elongate it from the 3' end this way, 3', yeah, this is antiparallel. These guys here, antiparallel. It works. Which means what for the other strand? Well, if these two are at the origin, and we know 2 are at the origin and 2 are at the fork, let's guess they're at the fork. So if we try this here, 5' to 3', is that antiparallel? 5' to 3', it's so beautiful! I'm fitting for clumped. And then DNA polymerase 3 goes to here. This is the 3' end. Yay, that is antiparallel to this 5'. That's this strand that it's complementary to. It works. This guy, 5', 3', add to 3' ends. Bingo, antiparallel. So happy. Makes me happy. Go watch the other tutorial. If you don't know what the heck I'm doing, it's all spelled out there. And now, my little friends, it's your turn. So do that. Do what I did. Don't cross the origin. You need your primers. You need your new strands. You need your 5' and 3' labels. Tell me which different colors for primers versus new strands. Arrow on ends, so I know which way they're going. And then upload to, I don't know, what should I name it, people? Let's just call it iPad number 28. I don't see a 28. 28 homework. Number 28 homework. Yes. 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 Ooh, what is this? This looks like I'm going to ask you to do some complementary base pairing. Which side first should we decide to put our primers? Again, antiparallel. And we want our 3' end not to be up against the origin, because remember, we cannot cross the origin. So you can use trial and error, it doesn't matter. But you can also sort of go, well, if this here is the 3' end, right, then a 5' has to be here, and it would go this way, because it has to add the 3' ends, so that means 5' here, 3' end. I think I will start over here with my primer. And obviously, I need to complementary base pair. I'm going to pretend like my primer is only 5 base pairs. That's the 5' phosphate of that C. This is the 3' OH of this A. That's my primer. I'm going to change colors when I start polymerizing. Then, right, and so forth, 3' OH toward the fork. Does that make sense? Yes. I'm just going to do 1/2. How about a primer for this side over here? AUCGG. Hey, why did she just put a U in there? Anyone? Anyone? Why is there a U? Because it's an RNA primer. It actually is a ribonucleotide. It's not DNA, it's RNA. You've got your RNA in my DNA. I know, weird, huh? But we don't have RNA in our DNA. We'll get to that. If you didn't watch part 2 of the tutorial, you're in big trouble, because now you don't know what the hell I'm talking about. Okay, anyways. But you'll see it. Go watch part 2 of the tutorial. We talk about that. How to get rid of the primers and it adds back DNA. Remember, we alluded to that. DNA polymerase 1. Okay, you're fine. Anyhow, then we add the 3' ends. And now we're re-adding. Come back, come back. Now we're adding DNA again, so there's T's, right? Whoa, holy crap, I don't even know how to complementary base pair. CCG. Stop at the origin. This is the 3' end. Complementary to this 5', 3', 5 and 3, 3 and 5, 5 and 3. It all works. That was crazy. Oh, and that's my blank slides that I accidentally. Aha! Homework upload. You knew I was going to ask you to do it. We're going to call it number 28.1. So fill this in like I did. You don't have to do all 4 primers, but at least do 2 of them. 1 for a leading strand, 1 for a lagging strand. And please label. You'll be glad you did. some definitions. This again, all our memorizing stuff, but we need to know it. So there you have it. There's the tutorial. Do your uploads. Read the book. Watch all the videos. Watch them again. Do the homework problems, end of chapter problems, quizzes, whatever. Go forth and prosper, people. It's good. You're gonna love it. It's great, right? Yes. Yeah, I know. You're not even Groot that time. You were kind of like half of a Groot.
Now, this is important.
Thank you. It is important. Malone, out.